parte de una defensa para formar un compartimiento duro, ayuda a proteger el árbol ante algún evento de contaminación, una lesión o incluso una infección. Por lo tanto, desde que entendemos el porqué de la presencia del calcio, podemos considerar que hay aquí alguna demostración, pues, del efecto que está condicionado el ecosistema de manglar como tal. Si podemos observar la distribución del calcio en la zona, en el área, de mayor impacto de derrames, hay un pico muy elevado, que es el mayor, de hecho, en todo el análisis que hicimos a través de nuestras rodajas durante estos periodos, lo cual de alguna manera podríamos correlacionar con el efecto de estrés generado por la presencia del petróleo. En el caso del potasio, según la literatura, es un elemento que de forma natural en la madera sana de una especie vamos a encontrarlo cargado o en mayor presencia en los últimos años o anillos de crecimiento de nuestro árbol. Si nos damos cuenta en la gráfica de abajo, la concentración de potasio es bastante estable durante todo el análisis de la rodaja, mientras que para la muestra expuesta a derrames hay un aumento o las mayores concentraciones de potasio detectado justo en el periodo de mayor impacto por derrames de petróleo. Entonces, también aquí podemos considerar que hay cierta efecto que puede estar siendo causado por la presencia de hidrocarburos. Recordemos, pues, que los hidrocarburos, la mayor problemática que causan es generar ambientes escasos de oxígeno debido a la capa impermeable que genera. Y bueno, otro de los elementos muy importantes que consideramos para analizar en esta investigación, pues fue la distribución del carbono, que en primera instancia, pues, está ligado al crecimiento natural de cualquier especie. En el caso de la madera, el carbono está presente en un 50% masa en la madera de los árboles. Sin embargo, podemos observar aquí como en la muestra de abajo, que es la no expuesta, la composición de carbono en el periodo de mayor impacto es bastante homogénea, mientras que en la muestra expuesta a los eventos de contaminación, la concentración de carbono presenta picos y inestabilidad que de alguna manera se puede correlacionar gráficamente con el tamaño o el ancho de nuestro anillo. Según Albaldao y colaboradores en 2016, se puede considerar que esta presencia mayor, si pueden ver, estamos hablando de rangos de hasta el 56-57% de carbono, puede deberse a un enriquecimiento de la madera debido a la bioacumulación del crudo como tal. Sin embargo, para que nosotros no correlacionemos erróneamente estos patrones o estas variaciones, también buscamos elementos que nos indicaran de alguna forma la presencia o el impacto de los derrames. Por ello, encontramos únicamente en las muestras impactadas por derrames azufre y molibdeno, y podemos observar que hay una fluctuación de estos elementos en el periodo de interés a la vida del árbol. Además, se caracterizaron los suelos considerándose por debajo de los límites máximos permisibles de acuerdo a la fracción pesada de hidrocarburos, pero podemos observar en las concentraciones elementales que el carbono es mucho mayor en las muestras contaminadas que en las muestras no expuestas a derrames. Entonces, hay presencia de petróleo que puede llegar a ser absorbido por las raíces del árbol. Como conclusión, bueno, no se observa un patrón de crecimiento definido a través del tiempo en las cronologías, porque son muy bajas, por ello se hizo el análisis dendroquímico. Y en las muestras analizadas del análisis dendroquímico difieren de que las muestras del sitio contaminado del no contaminado, y las tendencias de estos patrones indican el efecto de un factor de estrés que modificó la química de los anillos de crecimiento. No se debe identificar la presencia de metales pesados, pero se consideró el azufre y molibdeno como elementos indicadores de contaminación. Agradezco
Muy bien.
su atención. Gracias.
Muchas gracias. Tenemos en este momento una pregunta que te hace Francisco Flores Verdugo sobre si no tuviste problemas con la presencia de falsos anillos en la estructura lenticular en la dendrocronología.
Sí, claro, hubieron bastantes inconsistencias en la parte dendrocronológica. Obviamente estamos hablando de una especie tropical muy poco analizada en estos estudios, pero por ello estamos tratando de reforzar esta parte dendrocronológica con la química para tratar de validarlo, porque las cronologías pues se basan principalmente en la distribución del clima a través del año para el crecimiento del árbol. Entonces estamos tratando de reforzar esto con el comportamiento químico, la composición química del árbol.
Muy bien, vamos a pasar a la siguiente y última eh, ponencia de esta primera parte de la mesa de ecofisiología. El título se llama Condiciones ambientales y organismos asociados al cambio de coloración de la laguna salina bajos de Coyula en Oaxaca. Lo presenta Morales Flores Andeu Danay. Adelante, video.
Muy buenos días, es un gusto presentarme, soy Andewi Danay. Les voy a presentar el trabajo de titulación que llevé a cabo en la Universidad del Mar, se llama Condiciones ambientales y organismos asociados al cambio de coloración de la laguna La Salina Bajos de Coyula. Como sabemos, las lagunas costeras son un ecosistema que abarca aproximadamente unas 592 unas 125 a 130 lagunas costeras en todo el país. Aproximadamente eso quiere decir que están abarcando un espacio de 15,000 kilómetros cuadrados. Por lo tanto, es uno de los ecosistemas más comunes en nuestro litoral mexicano. Las costas de Oaxaca tienen aproximadamente algunas más de estas categorías de 1,450 kilómetros cuadrados, y pues nosotros sabemos que las lagunas costeras tienen una gran disponibilidad de nutrientes porque entendemos que al estar ubicadas en una parte baja de la cuenca pueden tener un gran arrastre de sedimentos y otro tipo de nutrientes, además de la materia orgánica que puede entrar al ecosistema. La remineralización bacteriana tiene este proceso de reciclaje, por lo tanto, los nutrientes tienen una mayor disponibilidad, por lo tanto, pueden producir una mejor productividad primaria en estas regiones, por lo que son unos ecosistemas con una gran complejidad y una mayor producción pesquera. Son ecosistemas altamente productivos. Por lo tanto, en este estudio quisimos saber realmente qué sucedió cuando la coloración de la laguna La Salina cambió de color, pues era una laguna verde y se volvió rosa. Estos estudios anteriores en otros países nos han dicho que hay organismos que pueden proliferar bajo ciertas condiciones. ¿Qué condiciones se presentaron en la laguna de Pruebos? En esta laguna de Pruebos, Caumet reportó que hubo una gran entrada de nutrientes, por lo cual hubo una excesiva aportación de algunos en específico que hicieron que la proliferación de bacterias púrpuras le dieran esa coloración. En cambio, para el Mar Muerto, Oren y Gurevich en 1995 reportaron que las aloarqueas eran las que le dieron esa coloración roja. Sin embargo, sabemos que en ambientes de alta salinidad, Oren en 2002 nos indica que las densidades microbianas pueden ser de hasta 10 a 7 células por mililitro, 10 a la 7 o 10 a la 8, por lo cual es una gran cantidad de células. Estos organismos halófilos los podemos encontrar en los tres grandes grupos de organismos vivos. Tenemos a las familias Bacteroidetes, Firmicutes de la bacteria, y las arqueas pues se encuentran principalmente en el grupo Halobacteriaceae, que son los que tienen esa coloración roja característica de estos cuerpos hipersalinos. Y pues este es el trabajo de Madigan que reportó para saber cuántos organismos halófilos podemos encontrar. Nuestro trabajo tiene este objetivo y se justifica gracias a que los organismos pueden tener una gran resistencia a la salinidad dependiendo de qué tan adaptados están. Por lo tanto, el objetivo principal fue caracterizar estas condiciones ambientales e identificar a los organismos asociados. Para esto, el primer proyecto se hizo de forma interna por Santiago Morales y colaboradores que trataron de tomar los primeros parámetros, encontraron una densidad celular promedio de 748.8 por 10 a la 6 células por mililitro en una salinidad de 50. Estas células tenían un diámetro de 1.7 a 2.1 micrómetros. Esto sucedió el 24 de febrero. Para el siguiente muestreo ya se tomaron los siguientes parámetros. La laguna La Salina es una laguna costera pequeña con un canal cerrado. En este momento todavía sigue cerrado. Es un canal de 1 kilómetro de longitud de ancho, 1 kilómetro de longitud y 90 metros de ancho. El espejo de agua puede variar dependiendo de la temporada, si es de lluvias a 52 hectáreas y si es en sequía a 20 hectáreas. Por lo tanto, la profundidad también se ve afectada drásticamente. El clima y el suelo de esta región pues se vio afectada principalmente porque el debilitamiento del evento Niño sucedió durante febrero y abril de 2016. En esa época, Oaxaca fue uno de los estados más secos y llegó a tener una temperatura máxima de 31.2 grados centígrados. Además, también se tiene que tomar en cuenta que es una zona con un cierto grado de erosión debido al tipo de suelo que es arenosol y regosol. El impacto de esta zona en términos de degradación ha sido de grado moderado a causa de la actividad agrícola. El muestreo se realizó con una botella Van Dor en 7 puntos y se tomaron muestras para análisis de nutrientes y parámetros físico-químicos. También se tomaron muestras frescas para el aislamiento y oxígeno y temperatura in situ con un multiparámetro GSI. La salinidad se midió con un refractómetro portátil en el laboratorio. El tester de pH se hizo conteo con cámara Neubauer, análisis de nutrientes en la Universidad de Colima, el aislamiento en medio Fenning, la tinción Gram, la tolerancia a la salinidad y también se hizo una extracción de ADN para posterior análisis externo en el servicio de Lambama y IPC. En análisis también se tomaron en cuenta la observación al microscopio, la descripción de células y colonias, los parámetros se observaron y se compararon también con los nutrientes. Los análisis de las secuencias de bioinformática serían posteriormente presentados. En los parámetros físico-químicos tenemos que es una laguna hipersalina, porque en realidad todo el cuerpo de agua tuvo una cantidad de sales de 51 a 53 en todas las estaciones. La condición del agua rosa, las condiciones eran que el oxígeno disuelto era de 0.2 a 0.62 miligramos por litro. Las temperaturas fueron de 28.5 a 31.3 grados centígrados y un pH de 7.65 a 7.89. Por otro lado, en la estación donde el agua era color verde, el oxígeno disuelto fue de 8.31 y 10.95 miligramos por litro. Las temperaturas fueron mayor, de 34.3 a 36.6 grados y el pH de 8.36 a 8.8. Los nutrientes tenían una cantidad de nitratos más nitritos de 13.64 a 23.84 micromolar y de amonio teníamos de 0.73 a 1.25 micromolar. Los fosfatos teníamos de 10.41 a 32.8 microgramos y de silicatos de 70.96 a 88.97 microgramos. De estos organismos aislados se observaron células de entre 1.1 y a 10 micrómetros. Eran organismos procariotas, 2 de ellos eran gram negativos y 1 gram positivo, crecieron a salinidades superiores de 2.5 de cloruro de sodio. Por lo tanto, concluimos que los organismos aislados pueden sobrevivir a salinidades mayores a 2.5 molar. Estas condiciones de hipersalinidad permitieron a los organismos mayor adaptados y con esa coloración roja que presentaron, pues ser los que predominaban en estas condiciones. Por lo tanto, se considera que fueron organismos extremalófilos los que le dieron la condición rosa a la laguna La Salina.
Perfecto, aquí estoy.
Por lo tanto, también se recomienda hacer un verdadero estudio de la hidrología de las lagunas costeras, ya que estos son verdaderos ecosistemas en riesgo. No se sabe, no se conoce a ciencia cierta la hidrología, porque sabemos que son muy cambiantes de acuerdo a las estaciones, pero presentan un riesgo a extrema salinidad, por lo que también se debe de analizar la distribución de la columna, en la columna de los parámetros físico-químicos y los nutrientes. Y se recomienda la realización de una secuenciación masiva en caso de que se volviese a presentar un evento de este estilo. Se agradece ampliamente a la Sociedad Cooperativa Bajos de Coyula, quien ayudó a hacer el muestreo. También mis colaboradores de la Universidad del Mar, y que ellos me dieron el seguimiento para poder presentar este trabajo de tesis. Les agradezco mucho, quedo a sus preguntas.
Agradecemos mucho tu presentación, Andeu.
Sí.
Hasta el momento no tenemos ninguna pregunta a tu presentación.
Sí, muchas gracias.
Por lo tanto, pasaríamos a la siguiente parte del Congreso con la presentación de la conferencia magistral. Muchas gracias a todos y esperamos en la siguiente sesión de ecofisiología.
¿Pausa el micrófono?
Sí, por favor.
Ok, ya estamos por comenzar la magistral.
Ok, perfecto.
Gracias.
Good morning, everyone. We welcome Dr. Dr. Lopez Portillo is a head researcher of the Institute of Ecology in ECOL, a public research center belonging to the National Science and Technology Council. He's a member of the National Center of Researchers Level 3. With students and colleagues, he performs research in different mangroves, the communities, the restoration and management. He coordinated the long-term monitoring program for mangroves supported by Conavio since 2011. He has been working in a hydrological restoration project in Tuxpan, Veracruz, supported as well by Conavio and by the UN Organization for Development, as well as CONAFOR. He's a member of the Botanical Society of America. He has been the coordinator of the National Wetland Community in the National Protected Area Association. This morning, Jorge Lopez Portillo will be talking about ecophysiology for the restoration of mangroves. Doctor, you have 50 minutes for your presentation, and by the end, we'll have 10 additional minutes for questions. Please go ahead, you may begin. Okay, my microphone is on, and I will start my presentation. Can you all hear me? Yes, we hear you. Well, thank you very much for the invitation to the organizing committee for having me in this Mexican Congress on the mangrove ecosystem. We want to have a comprehensive, integral knowledge for the conservation and management of the ecosystem. Thoroughly enjoyed this Congress. I have attended more of the conferences than in the live congresses, because now you can go back and rewatch certain concepts and in this virtual setting. So thank you for that organization. The topic that I will be presenting today is related to ecophysiology of mangroves as a tool for conservation. The content is related to the structure of the plants, the roots, the stems, the leaves. We know that roots exchange gases in a flooded and reducing environment, and the stems and leaves have a relationship with the interspecific hydric relationship, embolisms, stress, and the gradient of tolerance. The roots that are the support for the mangrove. Some of them are radial, most of them 50 centimeters or 70. In the roots, there's a specialization. Some of them are exchanging gases on the atmosphere, and there are also respiration processes and the important formation of rhizospora in this environment. There are also absorption roots, fine roots that translocate the oxygen from the atmosphere to the soil, and they also transport different organic compounds to the rhizosphere, and this is something that the organisms feed from. These places are where the ultrafiltration of salts is happening and where they regulate the osmotic and hydrostatic pressures. We've been already hearing about the amount of roots on mangroves. They cover like 60% of the biomass. And in arid zones, it's even 3 times more than the aerial biomass. This is a root and nematophorus. This is a radicular system with these fine roots, the pneumatophore and the nematodes. Here we can see Lagunculera racemosa. This is the species. These spots are the lenticels, the place where the oxygen is taken and water is released, and everything in red, making this cross-cutting cut. We can see the parenchyma. These are the roots. We have these cable roots, then we have the pneumatophores, then we have these absorption roots that are like a web. We have different sizes of roots, and here we have a representation. The xylem, and according to the diameter, it can transfer the different nutrients. This is a really small diameter, but in the cable roots, we see an increase in the diameter until it reaches considerable values. And we see how the diameter or the area of the xylem increases, and at the same time, conductivity also varies, the amount of water that goes through the root. These are the pneumatophores. These Xenia herminans, a quite dense forest, also speaks of the anaerobial qualities of the mangrove. And here you see that the density is related to a decrease in the sulfate concentration. That is the function, because they are like the snorkels that allow them. This is a block of a soil that is well preserved. We have these healthy pneumatophores with the lenticels. Then we see this organic horizon, which is quite significant, very dense, well structured. It has all of these things. These small things are the roots. This is a cross-cutting perspective. You can see the xylem here, and then there's a well or a clear boundary between the clay and the organic horizons. When we see the length of the pneumatophores, we can calculate the maximum level of flood that a mangrove can support. This is all the pneumatophores that are totally flooded, and this changes when we see a reflux or when the coastal lagoons are having differences. They open and they get closed, and there are changes. But the problem is when there's an interruption of the hydrological flow, and the water that stays there just gets stuck on the mangrove. And in that case, evaporation starts concentrating the amount of salts, and the reducing process on a root level is quite severe, and the oxygen contained in the pneumatophores that is accumulated is completely depleted. This is a block that has been decayed. We have seen that the pneumatophores are completely gone. You can see the holes here that disappeared. There is no evident parenchyma. This horizon is collapsed. There's no structure. We see the soil collapsing. This has really severe and serious consequences for the regeneration of the mangroves. This is a natural substrate. This is dissertation that has already been presented by Angel Zaragoza in this Congress. We see no seedlings, but in the artificial substrate, he installed these things that made them float, and they are like 10 meters from the soil, and the seedlings established there, and they look quite different. These are seedlings that have these short stems and really small leaves. And if we estimated the proportion of the stem and the roots would win, because these roots are looking for a substrate and they will keep on looking until their reserves run out or the amount of photosynthesis becomes insufficient. When that happens, we have these skeletons of seedlings in these areas with these secondary roots that are for non-absorption, and these leaves that play no role because they are really, really tiny. If we compare it to well-preserved areas, we see seedlings with a high density of fine roots and this anchorage roots and an important amount of leaves. In this experiment performed by Angel, it is interesting to see how the amount of propagules that got to these areas were around 70,000 propagules per hectare. Once they are set as seedlings, they lowered to 15,000. And the problem came when the season passed and the level decreased, and then the roots started having this issue and the seedlings died. This happened in two consecutive years. The sulfide measurements performed by Angel are a really shocking contrast. We see 5 milligrams per liter in well-preserved areas against 45 in decayed areas. This is a mechanism showing the importance of the rhizosphere. When we see a normal photosynthesis happening, the roots release this radial oxygen, and this oxygen oxidizes this sulfide hydrogen, and then the roots take the nutrients and they are sent to the leaves for performing the photosynthesis. And this also contributes to the radial oxygen. The opposite happens when we don't have a rhizosphere. How have we solved this problem in the restoration studies? Well, by increasing these aisles 30, 40 centimeters above the level with 5 meters of diameter. In September, we can see the situation, and then in November, we can see some plants, and in September, 1 year later, we can see high density. Unlike what happened on the other case, here we can see them surviving throughout the year. We are expecting to keep on increasing the density in these aisles. The height of these aisles, it depends on how collapsed the soil is. For instance, here in Alvarado, Aníbal Ramirez and his colleagues, also in Pronatura Veracruz, they have these aisles of 60 centimeters of height, 1 meter in diameter, and the resource ones help these little aisles to get stabilized and to expand. What are the consequences for the process? This is in the year of 2014. This dot is the same one that in the lower image. Here we did not have mangroves, and now this same regular pattern can be appreciated on the north and on the south, and it has disappeared because the canopies of the trees that have been established there are now touching one another. So it is difficult to recognize, at least in this satellite image, their recovered area. This case is in Pichavaran in India. This is a completely collapsed soil. Many attempts have been to recover it. And one researcher proposed, Selvam, he proposed to to elevate this substrate and at the same time to bring water so the substrate has better recycling process. So he made these two channels, these long channels that have like 3 meters of width and 1 meter of depth, and the small ones that have 0.75 in depth and 1.5 of width. This was done in 1996, and 7 years later, you can see some green areas, and now in 2016, you can see the density now, and the coverage is hiding the distribution pattern of the mangroves on these channels. These channels are also used by the fishermen for their catches and also for finding other sources of food. When it comes to the aerial part, we can see that the stem is important, is the mechanical support, and it provides the height so the canopy can expand. We know that there are short trees and that are extending horizontally, but it's always through the stem is the one that provides the distance for the expansion. And it also plays conduction roles in the xylem and also in the phloem of the sap that is elaborated. So the sap goes to the leaves and the other sap goes down to the roots, and the leaves become the transpiration and photosynthesis place. The photosynthesis not is just a mechanism of gas exchange and water for CO2. It's more like allowing or enabling a a pressure potential. This pressure potential is managed through the theory of transpiration cohesion theory. And this has to do with the fact that the water is taken from this root through the stem and then to the leaves. But the process that makes this possible is transpiration. These small leaves on top are the ones that are pulling the water from the ground that has a negative potential, sometimes has twice the salinity of the water, up to the leaves have a higher potential. This is a graphical uh, idea of what the mangrove has to do to pull the water from the ground and up to the leaves, the soil, the roots, the pneumatophores. And just to give you a scale, I put it in these sizes, and it has great potential. It's infinite. So this hydric potential is a key indicator, ecophysiological indicator. We can see this quite extensively in literature, and it's as simple as using these devices, these pumps, these pressure chambers. You cut one twig. When you cut the twig, it gives you this hole, which is proportional to the tension, because plants work under tension, and then we put nitrogen through the chamber until this notch, this indentation rises to the surface and starts making bubbles. That's when we can measure the water potential in the plant. And there's also a correlation between the water potential of the soil, the salinity. The water potential of the sea water is 2.4 megapascals. Here we have 12 megapascals. So imagine what that is when it comes to the salinity. And here is a stem. There's a linear correlation here, but observe that if we run a residual analysis, all of these points are above the line and just a few are under the line. This is the proportion 1:1. On the right, we can see Batis, and we have Conocarpus, and then another Conocarpus. Batis has this high potential. This is a plant with an enormous amount of salts to give these negative results. And Conocarpus, we can find it there. And then we have a full gradient of different species: Laguncularia, Rhizophora, Avicennia, Germinans. For me, it's the champion of all the species. And then we have Conocarpus erectus and Batis maritima. that are part of this community of water potentials. And this is the salinity of the sea water. So water potentials, as you saw earlier, it's during midday, and this is what happens with the water potentials before dawn. In orange, we have before dawn, and in red, it's midday, it's noon, and you can see 1 megapascal, it's equal to 100 meters below water. It's a really high pressure. Here we have 400 meters below water, and during noon, we have 6.5 megapascals. So the spread is quite important here. Rhizophora also has a wide spread, and if we compare it to sugarcane, it's nothing. The difference between potentials in pre-dawn and noon for sugarcane. So this capacity the mangroves have for differentiate between pre-dawn and noon is what provides the tolerance for that stress. So how can we measure stress? We have to see how these conduction in the coelom, if there's an embolism, then it is not possible to transfer anything. So perhaps these species that have a wider spread will have more tolerance to the loss of conductivity. And for that, we need to know how an embolism is formed. We have a hypothesis, which is the air crop hypothesis. And all of these holes on the walls starts forming. We start seeing these bubbles that are separated from the walls, and then they rise until they create the embolism. And once an embolism happens, then water has to go through other ways. But this gets completely covered, completely blocked, this vessel. And look, this is a twig without embolism. You can see the pigment that is covering the whole area. And with embolisms, we only see some vessels covered, and the others are completely empty. So water could not run through them. Look, the vessel, they are really, really to observe. And in order to determine that, we need to cut some roots, some twigs, branches. We have to calculate the length of the vessels. We have to cut down these twigs without without putting any embolisms ourselves. And we take these twigs and we use bury the place. We have the twig here. We enter the hydraulic pressure on top. And once it gets down, the air, you can see this is a highly twig with a lot of embolism, and these different representations of that segment. The diameter of the vessels is important. The species with the higher diameters are more resilient. No, the other way around. Are not. It's the other way around. For instance, Laguncularia is more fragile than Avicennia. This is a chart explaining the relationship between the different species and the conductivity, the specific conductivity against the percentage of the conductivity loss. Look, Avicennia herminans that has really small vessels has these loss of conductivity of 6 megapascals. It has a specific conductivity that is way lower than Conacarpus, for instance. And this happens not only between species, but within one species. This is Laguncularia racemosa. This is a dissertation of Claudia Paredes Lopez under these conditions of 0 parts per thousand, 10 parts per thousand, 25, and 50 parts per thousand. You can see the conductivity that decreases with the highest salinity. So we have the loss of conductivity due to embolisms, and this means that the more embolisms, the less conductivity we have. And this is what we found when we ran the experiment to determine that, we found that here we have the water potential. We start observing the exposition of the branches to the evaporation with the leaves. And then when they get to a water potential, we cut it and we put it through the device apparatus, and we start observing these curves of conductivity loss. 100% means that there's no conductivity at all, and you can see that these adjustments allow us to get to an indicator that perhaps might be the water potential, in which we'll lose like 50% of conductivity. In the case of Avicennia hirminens, that is at 5.7 megapascals, Lagunculera at 3.99, which is less tolerant, and Carpus 369, and Rhizophora in this case 6.5. We observe that these are different sites, so we also expect to see differences there. Maybe if we did it in the same site, we would have similar results. So this is the first thing that happens when there's hypersalinity in the environment. We start losing conductivity, we start seeing the roots dying. But when can you observe a saline shock effect? This happens when they are performing transplants from one side to the other, and the plants really need to settle. This is an experiment where we use these salinities of 110, 30, and 50 parts per thousand. And we ran this experiment for 30 days, but here we only analyze the first 8 days. So for a mangle that has a PLC of 6.5 minus 6.5 megapascals is dropping to minus 4 in water potential by noon. And this is way different if we compare it to the 30 parts per thousand or 1 or 10 parts per thousand. This is a shocking response. In the case of Laguncularia, in the fourth day, which was our last recording, because it lost all the leaves, and Avicennia got to minus 6 megapascals. This is the osmotic potential in the leaves. We observe similar patterns. It goes to minus 4. Laguncularia, once again, it died, and Avicennia got to minus 5 megapascals. Something that's interesting to observe is that there's a pre-dawn imbalance, and this explains why leaves end up like this. And it turns out that we calculate the disequilibrium, consider that this the water potential at pre-dawn minus the water potential of the soil. And if that potential at pre-dawn is minus 4, and in the soil we have minus 7, then the value is minus 3 megapascals. This means that there's a reverse flow. Instead of having water coming out due to the transpiration process, it is going to the soil. This means when we change one seedling to a freshwater site to a saline water site, what we are doing is that we are giving that water from the plant to the soil. The plant is dehydrated. So all of these positive values, for instance, here in 50 parts per thousand, by the second day, Rhizophora was already dehydrated, as well as Laguncularia, and the one that has a higher tolerance, it's always Avicennia hirminens. And here we even observe that it is being, the transpiration is happening at 1 part per thousand or 10 parts per thousand and up to 30 parts per thousand. The water potential of the substrate is related to the osmotic potential of the leaf. Do here is take a sample of the leaf, we smash it, and we get the total concentration of salts in such a way that we start seeing an increase in the experiment, the level of salts. So at the same time, salts in the leaf also increase. In the case of Avicennia, Laguncularia, and Rhizophora, in any of these species, the leaves are taking the salts and they are using hydrostatic pressure for making enough water potential to take the water from the soil. Something interesting here is the stomatic conductivity because it is proportional to transpiration because this is the product of evaporation and conductivity. So evaporation is the result of this process, but at the same time, it has to do with the photosynthetic rate. If we have these stomatic conductivities that are too low, then the photosynthesis won't be happening, which is what's happening in these three cases. The higher the salinity, the photosynthesis decreases. And of course, that we start seeing embolisms forming. And how do we know that? Because when we see the tension, when we input more salts into the experiment or taking into account the different blocks, then what we start observing is that the specific conductivity of the plant goes down. So this is not a consequence of the vessels that it has, but the consequence of having these embolisms on the vessels. Finally, it is important to see that plants usually in mangroves that are under stress, under water tension, the first things to die are the leaves, the upper leaves and then the inferior leaves, and finally the pneumatophores. And this speaks once again of this gradient of tension that we have been discussing. When we ran these experiments to see the relationship between the stem diameter and the maximum height of the branches, in blue we can see the trees that are in a good state of conservation, and in red we can see those trees that are already damaged. So we see a dieback. The branches of the upper canopy are dying, and the red spots are telling us for a diameter of 3 centimeters, we would be expecting not 6 meters of height, but 1.5 meters. The trees are getting shorter. That happens if the damage only happened once, then the canopy can recover from that. But here we can see a large tree, and the same thing is happening. The first branches to die are the upper ones, and the lower ones are still surviving. You can see the gradient, the water gradient that we thought before. So we can measure that water gradient from the soil, not to the atmosphere, but to the plant. Once for this, we can put the pneumatophore in the pump so we can observe these charts where we have the soil, the roots, the lower branches and the upper branches. This is the water potential. And there are some periods that are more favorable to maintain these potentials, and there are really terrible times. For example, here in September during rainfall, the difference is quite significant. So we have some periods, in this case, we should have more photosynthesis and higher productivity here, and the well-preserved tree shapes this line, which is quite clear to observe here the water potential from the soil and up to the upper branches. We see a difference of 2 megapascals, while the damaged tree has a difference here of 4.5, and then it gets to 8. It's like 4 megapascals of difference. So the water demand here is higher, and that is why the plants are dying more on the upper part than in the lower part of the tree. So this continuum that we were observing and that we might be documenting, we are finally documenting it here. So I would say that in order to try and understand what is the relationship between the ecophysiology and the conservation of mangroves and the restoration, I would say that ecophysiology is a key element that will help us do more than we expected. There are interesting phenomena like the secretion of salt on the lips. It's said that Abyssinia is very inefficient because it brings a lot of salt to the xylem through the roots. But we can realize that this salt is an osmolate, a cheap osmolate for the lip, instead of having pressure potentials that can cause severe phenomena the level of the xylem, for instance, the collapse of the vessels and the loss of conductivity. It is simply uptaking more salts and then regulating this concentration by bringing it out through these pores, as we can see here in the drops. So in terms of ecophysiology, I am really pleased to see that in this Congress, we observe different works presented on the leaf absorption and experiments with propagules. We have works that are now in studies working on Rhizophora mangle. We see interesting studies are quite enlightening. We see Rhizophora mangle. This was done by Julio Alberto. We can see the determination, hydroponic system by Perla Lerma, and then something that really excites me a lot, which is how the fruits, the seeds made in the plants, and they depend on the mother and plants to sustain their growth. And we can observe how these seedlings are able to do this uh, work by analyzing shape. This is a work by Gabriela. I want to congratulate all of them for their work. And with this, I want to thank the students that have collaborated with me in this project, Claudia Paredes Lopez, whatever we right now, Olga Bartolo, Frank Ewers, Guillermo Ángeles, and Rodrigo Mendez Salón. These are the students that have helped me this ecophysiology study. And of course, I'd like to pay tribute to Alfredo León Ramírez. He worked in restoration in Alvarado, Veracruz from the group Review MX. He had a accident and he passed away a couple of days ago. This was a really large team, and I feel really sorry for this. He explained the restoration process. He really gave really good representations. He is no longer, but we have all work there so we can use it. This is a leaf of black mangrove with the Chinese name. And of course that I would like to thank this work group that has supported us throughout this time. Recently, we have collaborated with Arturo Saldívar and Rosela Pérez Ceballos and Paloma Ladrón de Guevara from ATEC, and Rosella from the Institute of Science of the Sea in Isla del Carmen, and all of these group of people that has collaborated with us. Thank you. Thank you very much to everyone, and I am ready for your questions. Very well, let's move on to the questions. Patricia Ocaña is asking, in relation to the sites where the level of the soil was elevated, how can you ensure a permanent success in restoration if there's no water flow? Well, in those sites, it will depend on the site. For instance, in Tapamachoco, we leveled that with opening of channels, large channels, to connect the decayed part to the lagoon. Found some tidal effects there. So we are expecting to see these aisles working quite well. In the case of not a problem of low eruption, it was more a problem of overexploitation of these mangroves. That's why they came up with the idea of wall aisles, because the trees are growing on these cones that they create with the roots. So as you can see, they have developed quite well. And in Selbam, the last satellite image that I saw showed me that there's a loss of coverage there. But what we are expecting from all of these experiments on these isles, when they are well connected, we expect to see the roots, as in any kind of regular mangrove, to start growing on the sediment, to start 2-3 millimeters, which is the level of the water rising. And in this case, we have to consider that the isle is going to be compacted. So this is a challenge for the seedlings, but we are experimenting, and this is the way we can recover mangroves, because it is not necessarily an unrestored area, a place that's no producing methane or carbon dioxide. They keep on producing these gases. So what we have to do is just to restore them, or to start making a source instead of a sink. This next question is by Maria Teresa Rodriguez. It is not quite clear what the efficiency is of these aisles for the restoration. Well, these aisles, the group of Anibal and his colleagues, they use the same technique that they have in Xochimilco for making the chinampas or the aisles, putting some vegetal material. They make like these cylinders, 70 centimeters of height, and they start planting there. They used to plant some seedlings that they had in nurseries, and now they just wait. They notice propagules go there by their own, and they establish there. So once they have these areas, these are cylinders, so they might tilt to one side or the other and fall down. So what they have found is that when they have Rhizophora mangle starts building its own rice, and they anchor them to the soil, and probably through a translocation process of oxygen from the areas with the highest oxygen rates. So the roots start becoming productive, allows the tree to grow. There are trees that are 10 meters of height, and it is shocking. And the distance between the chinampas, the aisles, is of 5 meters. We want to bring them further away from one another because the canopies get really close. We need to give more space. And once the canopy is closed, then the conditions change. I hope that's clear for you. Yes, we think so. And now the next question. The pressure go from higher concentration to lower concentration according to the scheme that you presented. Well, the water potential is negative. So you have to, instead of making a positive pressure, you have to create a tension, a negative pressure. So it's like if we were with using a rubber band, pulling it, and I pull it to me, so I need to perform a work there, and that work is a tension, and it would be different if I wanted to push you. So the tension that is being generated is from the soil to the leaf. So there's an increase in the value, but in a negative sense. We have a water potential that's more negative. For example, in the bottom, we 4 megapascal soil. On top of the plant, in order to have an optimal photosynthesis, it requires 6 megapascals in negative. So the water can flow from the roots to the leaves. So if the value, yes, it's higher, but in negative sense. Jose Pedro Ramirez is asking if the lenticels are exclusive to the root, or can they be on the trunk or on the canopies? Yes, there are lenticels all over the plant. And it is interesting because these lenticels also serve as diffusion points if the plant have the possibility to move the embolism. I mean, where's a positive pressure than the pressure surrounding the vessels of the embolism, it can like squeeze that bubble, and that bubble might be able to leave to exit the lenticels. It's an interesting hypothesis that lenticels are a radial way trees have for breeding, and there are trees with a high amount of lenticels, and it's the same situation for mangroves as well. Question. Mangroves transfer oxygen sediments, and there's an increase of the oxic and noxic relationship. Do you think this happens with sulfate? Uh, do processes change in a microbial way? Yes, Francisco, and I'm sure you can explain it in better. What's interesting, phosphora, is that there's a gradient created concentration that is closer to the root, and it's more anaerobic the farther you get away from the root. Means that you have like an assembly of with different functions. Bacteria that can use sulfur and those that are exclusively microbial. Carlos Zamora is asking, how can you explain the presence of red mangrove with these yellowish and leaves? What is the cause? Is it genetic? We would have to analyze that. If those greenish, yellowish leaves are due to a stress, we would have to see what the explanation might be. Maybe there's a lack of nutrient, or it could be genetic. I don't know if you have seen that there are trees that use tropical that do not perform. They are like red in color, and this is a genetic thing. Sometimes they are like albino leaves. Maybe it's about nutrition. If we have reducting soils and the nutrients are not available for the plant, perhaps you can have some of those issues on the leaf. But I really don't know. Another question. We have seen works being presented that show that the reforestation of red mangrove can survive salinities of over 70 parts per thousand. Physiologically possible? Well, certainly it could be. It is physiologically possible because we have observed it, but I wouldn't expect to see 4 or 5 meters. Maybe they would be way shorter than that, but yes. And what's important here, the important thing to discuss when it comes to plants is that you have to use salts as osmotic compounds to regulate the pressure inside, and on the cells you need to have the possibility to produce osmoletes that do not damage the functioning of the cell and are facing that water on the vessels, and to make this gradient that allows water and nutrients coming from the sap to get into the cells. So if you have that capacity, it is possible. So perhaps in mangroves, in short mangroves, already explaining that nutrients play an important role, but also it can be a limitation. Jocelyn Villalba asks, have you found in an organism any reverse effects? Do you think this is a reversible effect that can be observed naturally in the plant? Well, this is very interesting, especially for mangroves, because the water potentials inside, I mean, in the branches of mangroves in general are very negative. And in order to have these embolisms, once the vessel has an embolism, then you have an atmospheric pressure. So you have to create a similar pressure so you can do it. In plants that have a root pressure, it's easier because the pressure that you are generating, it's positive. So you are just moving all the embolism, and in mangroves that hasn't happened. There are some publications and we have data that suggest that, but when we sent it to be published, accepted because they didn't believe it. And interesting because in the case of Avicennia herminans, for instance, you have the phloem inside the xylem. You have these sequential, and this might be the mechanism that will allow us to reverse embolism, that positive pressure that you need to extract or to dilute the gas again in the water of the xylem. It's a whole topic that we would need to run experiments on and demonstrate it. I think it does, but the radial growth is creating new xylems, so you would have to what the relationship is, the cause and effect, the version of one mechanism against the other. It's a whole thing. Same person is asking, is the increase of sulfur that increases the water potential, or which factors create the embolism? In the case of sulfur, what you have is a phytotoxin. The damage is in a metabolic level. And when it comes to salt, if you have an environment where the amount of sulfur is not so large because it is not so close to the sea, because sulfur comes from the sea, it's the source of sulfur. What happens is that the amount of salt you will be determining the water potential. If you have salinity, like in the experiment that I showed, that vary from 10 parts per thousand to 40 parts per thousand in a short period, then that creates a deficiency, a really high water potential that in the vessels that are formed in positive levels tolerated and the plants die. I have seen cases of mangroves that had a pulse of hypersalinity and then it got dispersed. And what you see is that branches grow with really small leaves. Very well. We have many, many questions. Giovanni Avila says, congrats, and he's asking, you have trained many generations of mangrove experts in Mexico. Would you recommend something to a student that is starting to study the ecosystem? Well, it depends on the discipline. Every discipline is essential. I think that the original knowledge generation is the reason of why science exists. He's interested in all the restoration processes. In that case, it would be important to learn about hydrology, biogeochemistry, to study the different gradients of the trees. But diversity, like Ilka Feller showed us, is really relevant as well. Everything is important. The bacterial and fungi activity on the roots, the fungi that are on the plants, the role that they play, anything, it's actually important. Any discipline, you would need to get good guidance on how much you want to risk, because making science is quite risky. Just a couple of more questions. How do you consider integrate ecophysiology in the communities that are performing these restoration projects? Well, that's an interesting, and of course, these communities might have a team that allows them to regulate the salinity on the soils, also to find equipment that it's easy to maintain and not that expensive, having the necessary devices to measure and to see if there are changes in the landforms or if a channel is blocked. If we have to open it, how are we going to open it? Maybe through a hose, a level hose, like Francisco is doing, or he was doing many decades ago. But the observation capacity of the persons in charge of the restoration, and based on the risk, you would need to, just like any other site, you need to have a diagnosis. And you need the devices for that, and that can be expensive. Francisco Flores is asking, we have built platforms and we have been planting red mangrove. Sometimes we use white mangrove. And why are we observing this behavior of the white mangrove if we planted red mangrove? Maybe there are seedlings, and I think you were the first one to start working on these soil elevations, and probably the contribution of the propagules of Laguncularia is higher than Rhizopus. And perhaps the type of substrate, it's a sandy substrate that's well drained, then that is the substrate that white mangrove prefers rather than the red one. With this question by Diana Jacqueline, what are the challenges in ecophysiology in this decade of restoration? As I showed you earlier, now that I was looking for information for this presentation, I didn't find a lot on photosynthesis or stress on an experimental level in this restored area. It would be to see that, what is the relationship between photosynthesis and carbon synthesis and the accumulation in the substrate as well. That's really important. The bacterial activity, making sure that it is being through photosynthesis, no, sorry, through ecophysiology to have an early alarm system. Those are challenges. And all the physiology, all of these things about whether the mangrove leaves absorbing water or not, if this contributes significantly to the water potential. So there are many things to consider: ecophysiology, restoration. Very well. On behalf of all the participants, we want to tell you that this was a wonderful presentation. Thank you, Dr. Lopez Portillo. Thank you very much for your presence in this Congress. Relevant, and that will be all. Thank you. Thank you very much. Thank you. I appreciate it. Goodbye.